A very good morning, all and one. This is Dr. Mushtaq Sheikh from VS College of Pharmacy, head of the department PharmChem. Today, we are over here with yet another session of Research Connect series version 2. In our last session, we had discussed about the story of pharmaceutics, wherein we talked about the research avenues and career in pharmaceutics field. And then today we have thought of going ahead with another important area of research in pharmaceutical sciences, that is quality assurance and analysis. So student, many of you would be aspiring to be into this particular field of research and work. And definitely, needless to say, by this time, when you are searching for research career in pharmaceutical sciences, you would be aware that QA is a very important stream of pharmaceutical sciences, and no company can survive without this department. There are a lot of depth, lot of things we have to understand. So we have a very renowned person today with us. As they say that you have to hear something from horse's mouth. So today we have a very renowned person in the field of quality assurance, Dr. Atul Shirkhedkar sir. Sir is presently working as a professor, vice principal, and head of the department. I welcome sir with my hands folded. Welcome Atul sir. It was my long desire to witness and meet you. Welcome to VS College of Pharmacy's platform, wherein we are interacting with our students who are thinking of going to research area in quality assurance in particular. I will continue with the introduction of sir, though sir does not need any introduction as a formality. That's my duty. So sir is, as I was saying, professor and vice principal at R.C. Patel of Pharmaceutical Education and Research, Shirpur. He has a vast experience of 25 years in teaching, and out of which 18 years research and administrative posts. So he has hands in all the three directions: teaching, research, and as well as administration. Presently, he is also a member of Board of Studies of Kavitri Bahinabai Chaudhary, North Maharashtra University, Jalgaon. He has been on the board since a very long time, since 2011. Sir has completed his BPharm and MPharm from Department of Pharmaceutical Sciences, Nagpur, and he is postgraduate from KBC, North Maharashtra University, Jalgaon. His areas of research include validation of analytical methods, impurity profiling, bioanalytical method development, solubility enhancement, and phytochemical investigation for various formulation. He has supervised around 75 students for their MPharm dissertation, and he has guided 2 research scholars for PhD. And presently, there are 7 students who are pursuing research and PhD in his guidance. This is not just enough. He has shown his caliber of research, in research and innovation by publishing 170 plus research publications. This is not a small thing, and they're all journal of national and international repute. With this, he could have H index of 90, which is a huge number, and ITN index of 49. And mind well, his paper has been really been popular, and it has been cited around 1600 times. This is a huge thing to witness over here. Sir has authored 5 books in pharmacy and 9 book chapters. And this is not enough. He has even published 4 patents. So what I was saying was absolutely right, that we are hearing this particular guidance for QA and analysis area from Sir's very own experience. Sir has faced a lot of grants for research as well as for conducting workshop. For particularly second thing, that is conducting workshop, seminar, FDPs, he could gather 50 lakh rupees from various funding agencies like UGC, AICT, ICMR, KBC, North Maharashtra University, and he could himself organize 15 national conferences and as a convener and 25 conferences as member of organizing team. He has been honored several times best teacher by KBC, North Maharashtra University. has received Research Excellence Award for publication from TBC North Maharashtra University. He has also received a very well-known award called as PD Sethi, Dr. PD Sethi Annual Award for Best Paper. He has delivered more than 50 invited lectures in seminar and conferences, and also very active as a lifetime member of FETI and IPI, and been reviewing a lot of papers from reputed journals. So students have been really must be really convinced, sir, that they are going to get good guidance from you. I am also backstage, will be witnessing this uh, knowledgeable session from your end, sir. I invite Dr. Atul Shirkhedkar, sir, on the forum. Sir, forum is yours. Over to you, sir.
Uh, Thank you, Dr. Mustaq, for nicely introducing me. In fact, I'm really thankful to all of you for inviting me on this wonderful platform. In fact, I wanted to visit your institute in person, the Vivekananda Education Society's College of Pharmacy, and it could not possible because of so many reasons. But in future, definitely, I will visit the institute one. I extend my compliments and congratulations to you and your institute one for getting the CI ranking, that is the second position one. And along with that, I wanted to convey my regards and thanks to the principal madam, Dr. Supriya madam, then
Sure, sir.
the coordinator for this program, Hirlekar madam, and then Mushtaq, you yourself, and Anita madam. Plus, I am really thankful to my dear student, Ojas Agrawal, who is associated with your organization and doing the wonderful things at the institute. Really thankful to all of you for giving me an opportunity to share my thoughts and views on this occasion one. And it's a good, you may say that the wonderful activity they have started one. So compliments to all of you for starting such type of the uh, innovative practices one to bring all the students, academicians on that of the one platform one.
Yes.
And I can say that even though I am 450 kilometers away from this your place one, but at the same time I'm thinking that I am in the Mumbai only with your in your organization, right? So thank you. To begin with, you have introduced me in the very good way, and I hope, I don't know I am deserving this or not, but okay. Thank you. This is what I wanted to share with the student. Dear students, you are joining this, you may say that of the program. I compliment you for joining such type of the activity one. And today's topic that I'm going to present, that is the research avenues and career opportunities in quality assurance and analysis one. Basically, I am the person from that of the pharmaceutical chemistry analysis, quality control, and that of the quality assurance one. So you may say that of the from the very beginning one, I keep my interest in this type of the subject one. This subject will be serve the purpose of that of the industry, and I hope the probably whatever the things that I could gain as a, you may say that of the experience one, that I wanted to share the experience with my students also, and so that they will be the comfortable in their career one. I hope to begin with this, the first slide I wanted to share this one. That is a success mantra for that of the you all people. That is, you can take up one idea. That is very important one. Where you wanted to go, you choose one idea. In the Vivekananda's quote, I can say, make that idea, one idea, your life. Think of it, dream of it, live on that idea. Let the brain, muscle, nerves, and every part of your body be fully full of that idea, and just leave every other idea alone. This is the way to success, dear friends. I wanted to say that with this objective only, you should start with the dreaming. You should start with that of the, you may say that of the thinking of it, and probably you can achieve whatever the dreams that you have. You can achieve that dreams very soon. And the next, the slide, while going with that of the next slide, I can say another important aspect that I wanted to share with all my students, those who are participating over here, keep moving and keep going. This is what important thing is, that if you can't fly, then run. If you can't, if you can run, then walk. If you can't walk, then crawl. But whatever you do, you have to keep moving forward. This is what is very important one. And the student, those who are thinking that I can't, you may say that of the work on that of the instrument, I don't have that knack one, or you may say that of the expertise one, or at the same time I don't have the confidence to operate the instrument one. Probably such student should try to go with this one. Whatever the things that you are doing, try to enhance that one. If you can't work, then I wanted to say that sometimes you try to go beyond that, try to stretch yourself and do the best. So, dear students, the very four important areas in that of the pharmaceutical industries, one, four or five areas I can say. But from your point of view or from my point of view, this is the first area which is very interesting to all of us. That is the research, the purchase of raw material one. We never thought about it. That is it the area in that of the pharmaceutical industry? Yes, because raw material is required, because API is required, excipients are required, solvents are required to manufacture the things. And when you must say that of the purchase department is procuring such type of the raw material one, I think they are having the quality control department related to it, and whenever they are purchasing that one, they are trying to see whether it is of that quality or not. Means purchase department is one where you can say that of the you can have an opportunity over to, you may say that of the to get the jobs in that particular section one. Second important area, you can say the production or the manufacturing one. That the production or manufacturing one, the basically the most of the time the student, those who are having the, you may say that of the bend of mind related to that of the manufacturing type of the manufacturing or designing that of the formulations one. Well, it may be that of the tablet, capsules, oral liquids, parentals, and all that one. You can have that different sections one, and in that different sections one, you will find lot many students, or you may say that of the employability is there for that of the, or opportunities for that of the SUTICs and the QA people. But at the same time, the IPQC department is one where you can say that of the you can find out here the area of interest. Similarly, the you can have that is the quality control and that of the quality assurance department one. And nowadays, the people are thinking about the quality. They are more cautious about that of the quality, and they wanted to assure that one also. They are not satisfied only with that of the quality one, but at the same time, there is a need for that of the assurance of that of the quality. Then finally, you are going for that of the packaging of that of the material that you are having, and then lastly, it is going to that of the marketing. But in between that, when the drug has been introduced into the market one, or you may say that of the, there are many issues related to it. Well, it may be that of the stability issues of that of the drug. Well, it may be that of the many areas are there, and that all areas can be covered up in the, you may say that of the research and development. Whether the development of formulation one or whether the discovery of that of the drug, these all can be covered into that of the research and the development one. So in this area, I think you have lot of opportunity one. You may say that of the, you have to just think that whether I'm satisfying the needs of that of the industry or not. If I wanted to go in that of the research and development area, everybody think they have to be there in the glamorous field. And in the pharmaceutical field, you can have an opportunity that glamorous field may be the research and development one. That glamorous field may be that of your the quality assurance one, quality control and that of the quality assurance one. I hope this quality control and quality assurance one or research and development fields are very important for all of you. Are in that particular area, you try to see where you may say that of the prove your metal, where you can apply your the knowledge of what you have gained during that of the during the 4 years, or you may say that of the 6 years of the graduation and then post graduation one. You can directly go to that industry and you can apply that knowledge in the industries. I wanted to convey the message that this is a very challenging area, and those who wish to take the challenges in their the life, probably that that people only can you will get an opportunity and they can do the best in their career one. I know that you all people are the very wonderful people, and they wanted to you may say that they go to the industries one. They wanted to do the lot of research work in the industries one, and I know the because of that, the probably you will be much more attracted to the field of that of the research and development, at the same time the quality control and quality assurance. Now try to understand when you go to that of the production section one, that time you know that something you are going to have in the SUTICs. I wanted to say what is important from your perspective one. If you are the chemist or if you are the analyst one, from your perspective or from your point of view, when you are manufacturing any formulation one, there are two important things. One thing is that is a raw, you may say that of the drug, it is called as the API, active pharmaceutical ingredient one, and the second part of it, that is the excipient one. The two things are very important. One drug, or you may say that of the API, active pharmaceutical ingredient one, and the second, that is a you may say that of the excipient. Both needs to be you may say that of the analyzed one. You have to analyze, you have to confirm that one, whether that same excipients you are using it or not, whether you are using the same API, whether it has been whether that API has been changed or not, that you wanted to assure first. And so how you are going to assure? That is, you are going to assure with that of the, or you are going to go with that of the quality control and that of the quality assurance side one. You wanted to confirm that all the things. Now, when the raw material is there in that of the purchase department is procuring some raw material one, they wanted to see the quality. They wanted to go for that of the quality control things of it. They wanted to check the purity of the compound. They wanted to see the assay and all that one. That all can be the same melting point, and then many things they are carrying out in that section. Now, with this, you may say that of the section, the production is going to be there. With that assurance, at the first stage, you are going for that of the production department one. You are going manufacturing certain things. In the IPQC department one, you have many opportunities one. But at the same time, I will focus attention because there are many students from that of the pharmaceutical analysis and you may say that of the quality assurance one. What opportunities they have in the pharmaceutical industry? I know this is the big section, because even if you have design, even if you have, you may say that of the develop some formulations one, unless and it is been quality control and assured one, it will not go to the market one. Means what? Your role will be such that once you are making the sign, that product will be there in the market one. Otherwise, you can hold the product because of so many reasons. Nowadays, you may understand that the people are talking about that of the impurities one. Instead of purity, they are going beyond it. They wanted to see what type of the impurities are there into that of the particular formulations one, and to what extent that impurities are there. What is the threshold level for it? If it is crossing that level limit one, they wanted to say that they cannot launch the product in the market, or it will not go to you may say that it will not be there in the market one, the pharmaceutical market. So quality control or the assurance related to it is a very important aspect. And the broadly, that is the packaging is there, which is also the very important aspect, where you may say that of the analysis will be there. Take example of, you may say that of the parental things. You are using the glass bottles, or you may say that of the plastic material over there, or rubbers also you are using. You should know how to analyze that. You should know whether there will be the leaching from that side one, that you wanted to confirm it. And who can confirm? You are the people who are going to confirm the smallest amount of that of the, you may say that of the material which has been leached into that of the formulation one, that you can confirm it. As I was talking about some sort of the impurities, yes, impurities are very important for all of us. If it is crossing that limit one, it will not go to that of the market, or the product will not be there in the market one, or it has to be withdrawn from that of the market, even if it has been found during that of the, you may say that of the, if the during that the product is there in the market. So I can say that overall you have the ample responsibility, big responsibility on your shoulder, and you have to, you may say that of the, with that responsibility, you have to given with that of the authority to sign, to pass, or to fail that product one. And finally, the marketing and all that one is area, but in the marketing one, your job is very limited one. I can say that the analytical people or the quality assurance people may have the job opportunities primarily in the area of that of the purchase of raw material, where quality control is also necessary. In the case of quality control and quality assurance department one, you have the wonderful opportunity one in the production and manufacturing one. And next, the glamorous area, which from the student's point of view, that is the research and development. In that area, you have the opportunity. So one by one, we'll try to move. When you are talking about that of the research, which is a glamorous field, in the research, that is what is research? This is a systematic process, because when the student is there in the B.Pharm level, they wanted to have acquire certain skills. They wanted to get the knowledge for that of the certain subject one. Take example of pharmaceutics, chemistry, analysis one, pharmacognosy one. You are learning certain things from that of the subject. At the same time, you are getting the experience, the practical experience related to it. That is how to get the sections in that of the pharmacognosy one, or you may say that of the analysis of certain formulations one, that you are learning in the analytical side. Even the quality control also you are doing at your the undergraduate practicals one, and these things are going to be there going to be helpful to you in your industrial career one. But in the area of research and development, which is a very promising area for all of you, that is what is research? That is a systematic process of collecting and analyzing the information. What you are doing? you are trying to collect the information and analyzing that information in order to increase our understanding of the phenomena with which we are concerned or interested one. हम लोग जिसके लिए concern है या interested है उसके लिए हमको जो data है, that data that we have collected, that we have to understand it, that we have to analyze it. This is what nothing but the research one. Then you may say that of the step by step you can say how you can start with that of the research procedure. This is first, that is, you may say that of the research problem. You have to understand the research problem. But how you are going to understand the research problem? Sitting in the lab only, can you understand the research problem? No. Sitting in that of the, you may say that of the, you may say that of the, in the air conditioned room, can you get that doing some meditation and you will get the problem that is, yes, this is the research problem one, and somebody will say that, yes, this is research problem. Now you try to go and solve the problem. Second, many times the students are going to that of the literature survey. Sir, I am doing my literature survey, and through that literature survey only, I am going to get the research problem. Now, try to understand where there is a problem. You have to go to society first. What are their problems? You have to find out what problems they are facing. One, whether if the formulation is not stable, so you have to understand that if the formulation is not stable, then how I can make that formulation stable one. That is the problem for that of the formulation one. If you may say that of the analysis for that of the drug is not up to that of the mark one, what are the opportunities for me? How I am going to solve that problem? Means what? That if you wanted to get the research problem one, the literature is necessary. Literature is important one, but beyond that, you have to consult. You have to have the observation first. That is, whether which type of the problem these people are facing one, and based on that of the problem, you are trying to search some sort of the literature one, whether somebody has given the solutions to that of the problem one. If they have not given the solution to the problem, then you have an authority, you have an opportunity to give the solutions to the problem. Then again somebody may say that if the solution is available, then whether that solution is affordable, whether that excellence is there, whether it is affordability is there, that you wanted to say. And now in today's world, you need to have excellence with that of the affordability. It should not be the costly one. It should be the easily available, readily available, and it should be the very simple solution to that of the complicated problems. This is what very interesting things to all of us. We never thought about that is the easy solution for that of the complicated problem. We wanted to make it more complicated one, just to have thinking in mind that is research is a complicated subject. Research is not the complicated subject. It is giving the solution, easy solutions to the complicated problems. So even though lot of efforts are required during you may say that of the all these studies one, but at the same time you can come out with that of the very easy solutions related. Take example of that of the, you may say that of the diabetic strips. That diabetic strip that you are using, the sugar level you wanted to check, just with that of the one prick, you can put that the drop of blood and you can see that is the sugar level one. You can monitor the sugar level. What does it mean? Means what? It is a easy solution. Initially, you are going to the pathology laboratory, withdrawing the blood. That are all steps now been, you must say that of the no need to perform all these steps. Right now, easily you can get with the one prick, you can get what is the sugar level, the blood sugar level you can check. Similarly, by that of the BP apparatus are also available one. Many things are there. So this type of the gadgets which are available in the market, these are all easy solutions for that of the complicated things. Now you can monitor it. That is, what is the blood pressure level, what is the sugar level, everything at your place only you can check, you can monitor that things. Now this is what in the research, they have done the research one, and they have got the easy solutions for that of the complicated problem. How they have, you may say that of the understood the problem, they reach to that of the old age people. They discuss with that of the patients one, and what are their problems, and they discuss with their family members also. Now I wanted to say that when you are discussing with the patients and their family members one, you will come with that of the problems related to it. I will give the one more interesting thing to you, that is many times the patients are missing the dose. They are not remembering it, whether they have taken the tablets or not. So in that case, do you have any solutions for it? You try to find out the solution, easy solution for that of this problem. Many times it happens, or sometimes there is a possibility of repeated dose of that of the drug also. So just to avoid that one, try to find out some sort of the, you may say that of the sketch, some sort of the design, some sort of the you may say that of the application one, where the patient will be the comfortable with that application, and at the same time the patient can recall or remember whether the patient he has taken or she has taken the medicines or not. that can be recalled. So this is what is an interesting one. If you are reaching to the patients, if you are going to that of the pharmaceutical industries, if you are visiting the industries one, if you are making your observation perfect one, you can come out with that of the research problem. Many times it happened when the students are going to that of the M-Form one, we are asking him that what topic you wanted to select for your the project one. The student says that, sir, since you are working in the analysis one, I wanted to work only in the analysis one or in the synthesis area one. Or somebody wanted to say that I wanted to go with that of the pharmaceutical formulation one. Now there the trend is there for that of the nano formulation, so I will work on that of the nano formulation. I never thought about whether there is a possibility, there is a need for that having the nano formulations for this type of the medications or not, or the drug or not. But beyond that, the people are trying to make such things just just to complete their project one. Dear students, do not try to complete the project. Try to give the solutions to that of the problems one. Even if the smallest thing you are doing in your the lab, that is very interesting one and important from that of the pharmaceutical industry point of view. So try to understand that you are when you are going for that of the M-Form one in that of the pharmaceutical chemistry or pharmaceutical analysis one or quality control one, you are the people, you are the authority who is trying to give the easy solution for that of the analysis one. And whatever the things that you are doing with that perspective only, you have to work in that of the pharmaceutical laboratory as if you are working in the industry one. Many times it happened that students are doing the practical in the laboratory and the college the faculties are insisting that you should have the logbook, you make an entry of the name in the logbook one, but never the students are making the entry in the logbook. Why? Because they have the fear that something gets wrong and the college authority will scold me that yes, the disturbance is due to you only. It is not that one. Try to understand that this is the documentation that you are learning at your, you may say that of the postgraduate level or even in the undergraduate also, the whatever the instrument that you are operating, make entry into that of the logbook. During your the lab work, you try to have the apron one, wear the apron. It is the status symbol for you that yes, you are working in the laboratory one, and with that apron it can give you the confidence also. It can give you the confidence also. So try to understand this is the small steps that you are taking heading for that of the research. It is not, you may say that of the something which is not beyond your capacity. It is there within your reach only. The research is within your reach. Only thing is that the step by step you have to go, you have to find out the solutions for that of the problems. Now when you are trying to go with that of the literature survey, review of literatures and all that one, you are referring the different journals, you are collecting the information from that of the different journals and formulating your hypothesis based on that of your the journals one. Now with that formulation of the hypothesis one, you are trying to go with that of the you may say that of the problems one, and you are trying to design that this is the problem, this is the hypothesis, on the basis of that I am trying to give the solution for it. Now preparing the research design, उसके basis पे आप उसका design prepare करेंगे, then data collection will be there, data analysis will be there, and interpretation of the data will be there. These are all steps you are going to find out because these are all the name you are trying to give. But in the pharmaceutical field, take an example that is in the pharmaceutical industry. If I wanted to develop the method for that of the particular drug, what step I am going to involve into it? Whether you thought about it, take example of your the paracetamol. Now I wanted to develop the method for the paracetamol. What is the possibility for me? Now it is there with that of the IP. In which case I am trying to develop the method for that of the particular formulation. Have you ever thought about it? Whether one method is sufficient for that of the analysis of that of the paracetamol, whether there is a need for that of the another method, that all things should come to your mind. Now you try to understand there are many analytical methods available for that of the one drug. There is a possibility that you can have for one drug nearly 70-80 methods are available, analytical methods are available, but the purpose is different. Now in what cases you are trying to develop the analytical method? The first case, that is if the pharmacopoeial method is not available, if the drug is new and no method has been reported in that of the image that of the literature one, and the pharmacopoeial method is not also available for that of the drug. In such case, there is a possibility, or it may you may start developing the analytical method for such type of drugs. The second case is that you can say that yes, there are the established methods, there are 2 to 3 methods available for that of the particular formulation one or particular drug one. So I wanted to develop the new method for that of the drug and that of the formulation one. Now in that case you will understand that is how you are going for that of the development one, that existing methods are having the certain drawbacks. they have certain limitations one, and they are, you must say that of the taking the lot of time for that of the analysis. In such case, you know that there is a, the, you must say that of the lot of things are involved, the time required for that of the analysis is more, many things are there. Now you wanted to work on that of this particular aspect one, how I can reduce the time, how I can reduce that of the cost of analysis one, that how I can reduce the solvent cost also, that all things I wanted to understand. The existing method is not that much precise one, so I will try to develop the method which is a more precise than that of the existing method. So this is the second, you may say that of the way in which second condition in which you are trying to develop the method. In the third case, you can say that if the impurity profiling is there, if the impurities which are present into that of the drug, if there is no you must say that of the availability of that of the method for that of analyzing the drug in the presence of the impurities, or analysis of impurities one, in such cases one also you can develop the method. This is all you can say that of the next stage that you can say. And the last, you may say that of the bioanalytical method also you can develop. If the availability of that of the bioanalytical method is not there, then you can develop such type of the methods. So this is related to that of the development of the method, when you can develop the method. But dear students, try to go to that of the pharmacopoeia first, because we are missing, we are not going to that of the pharmacopoeia, we are not referring the pharmacopoeia. What are the contents of the pharmacopoeia, you try to know. First stage, first thing is that in the, when you are referring the pharmacopoeia, you will try to find out, that is if you are going for that of the monograph for that of the any drug, you will try to find out that how they have identified it, what is the melting point for that, what is the boiling point for that of the material one. Then you can say that of the many things they have given into it, the chemical name for that of the drug, solubility of the drug, and identification related to it. Then you may say that of the characterization part of it, what type of the impurities are present into it, and finally they have come out with that of the assay, by which way you are trying to make an assay for that of the drug, analysis of that of the drug, that is also been given. And lastly, the what is the, you may say that of the applications of that of the drug, that is also been mentioned into that of the pharmacopoeia. Means what, in every case, when if you wish to go to the pharmaceutical industry one, you should try to go to that of the pharmacopoeia first. In your interview, I will give the certain tips to you, that when you will go to that of the interview one, you read first that all the pharmacopoeia, that is what is been given. I don't want it to say that the mug of the pharmacopoeia, but try to see which is the latest edition, what are the sections available in the pharmacopoeia, what type of the different methods they have given in the pharmacopoeia one. You try to learn that one. In pharmacopoeia, if you are observing, you will try to see that initially in the 65 and you may say that of the 85 pharmacopoeia one, you will see that is there is a possibility of having analytic method by using the spectrophotometer, because prior to it most of the time they were going with that of the trichromatic methods of the analysis one, then they switched to that of the spectrophotometric methods for the analysis, and later on they have developed now the recent pharmacopoeias which are there. You will find in the most of the pharmacopoeias they have, you may say that of the either HPLC method, GC, HPLC, or many advanced instrumental methods they have included in that of the pharmacopoeia. Plus they have mentioned the what are the different types of impurities which are present into that of the drug, that also been mentioned into that of the pharmacopoeia. So dear friends, try to read the pharmacopoeia by heart, try to see it, what are the different content. Even we are missing certain things in that of the while reading the pharmacopoeia. You try to see how you are going to calibrate the certain things. Now you know that you are doing your practical in the laboratory. You know, first step is that you are the glass apparatus which are there. Now, well, it may be the volumetric glass, well, it may be the pipette one, whatever the glass apparatus that you are using, whether that apparatus are already been calibrated. Have you thought about that calibration of it? And here, the role of analyst, role of that of the quality control person is there, that you should know how to calibrate the apparatus. What are the methods for calibration of that of the apparatus one? You should try to know whether how you are going to calibrate the burette, how you are going to calibrate your the pipette one, volumetric class one, that you should know. Now for calibration of it, mass upon volume, very simple things. You can say that of the mass upon, by using the mass upon volume, you can density is equal to mass upon volume, and with that you can say that of the, what is the mass of it, mass in the gram. Now you have the volume also. So if you know the density one and the volume one, you can calculate the mass related to, or you know the mass and you have the density, you can calculate the volume related to it. If you know the two things, the third things automatically you can calculate it. So the calibration of apparatus is there. But prior to it, the cleaning of the apparatus is also very interesting aspect one. Many times people are missing, that is how to clean the apparatus one. Now for cleaning the apparatus one, you try to see that what are the steps involved in that of the cleaning. There should not be the residues related to that of the there should not be the residues in that of the glass apparatus one. Otherwise, it may interfere, it may change the pH of that of the solution that you have prepared one. The pH of the solution may the change one. So in such cases one, you should try to see that is how you are trying to keep cleaning of that of the apparatus. Dichromate solution, if you are using it, whether you are using that is the detergent, water, how you are drying it, that all steps you are trying to you must say that of the involve into it, and you are finally drying the apparatus one, and then you are using it for that of your the day-to-day experimental purpose. So this is what the in the industries are following. They wanted to assure each and every step, and I know that you people are going to do the wonderful jobs in that area one. Now there are ample opportunities. You see, try to see how many pharmaceutical industries are there. Small and large scale industry, you can say that more than 35 to 40,000 pharmaceutical industries are small and large scale. I am talking about one where there is a huge requirement for you people and the area of, you may say that of the quality control, quality assurance one, and analytical side. You have the ample opportunities one. If you wanted to go with in that pharmaceutical section, plus you have the opportunity in the research and development also. If you wanted to go over there, what is needed for you, that is the skills will be required, certain skills will be required to you, and in this the skill needed in that of the quality control, quality jobs in that of the pharmaceuticals one, that is the GMP knowledge is required. Then quality culture mentality, keen observations and attentions to the details, and the problem solving skills, and excellent communication and literary skills is also required. So the two important skills, I can say that the one that is the soft skill is necessary, that is interpersonal communication is very important, because many times the people are you may say that of the facing the audits, and when you are facing the audits, it is becoming obligatory that you have to communicate to that of the authority and the inspectors one, that yes, you are facing the audit, so how, which type of the method you are utilizing it, how you have performed the analysis one, that all things you wanted to study related to it. Now when you are going for that of the, and you may say that of the authority, discussing that authority, there should not be the ambiguity in your the language, there should be the clarity, and that clarity will come with that of the confidence. And when there will be the confidence, when you are handling it properly, when you are doing that all the analysis in the proper and the systematic way, then you can say that it can build the you must say that of confidence in you. Now communication skills, for that you have to go with that of the soft skills and all that one you can learn. Your school and your college may I think is taking a lot of efforts in that area one, and you people can develop that, or you can make an improvement in that area one. But at the same time, the another important aspect, that is the technical skills, and which is very important one, very necessary one. Now what skills technique, when I'm talking about the technical skills, take example that as I quoted first, that is the glass apparatus need to be cleaned properly. Yes, it is very important one. Second important aspect, that is I wanted to analyze the certain things, so I will have to take the weight of that of the active pharmaceutical ingredient or the drug. Even during my practical also, I have taken the weight of that of the sample one, or you may say that of the drug one. When I took the weight of that of the drug, whether that weighing balance was calibrated one. Have you thought about it? Have you checked that the weighing balance was calibrated? Now try to see if there is an error in that of the weighing of that of the sample, how you are going to get the correct result. Many times it happened that students are saying that, sir, we are not getting the result, we'll write as it is, whatever the things, and they are doing the essay for that of the many thing, and they are having the reading, that is the marketed formulations, or you may say that of the, even the laboratory mixture they are preparing one, and they are having the essay result, that is 60%, 70%, 75%. Why it is so? Because they have not properly extracted the sample, they have not properly weighed it. There are many things, many errors have you may say that of the encrypted into that of the steps one, that they wanted to avoid it. As I'm talking about, that is the weighing is the first important stage in that of your the analysis one. So have you thought about that, how to you may say that of the calibrate the weighing balance? There are the electronics now, the weighing balance are available. Now if you wanted to calibrate that one, how you are going to calibrate it? There is a need for that of the internal calibration and that of the external calibration. If you are going with that of the different weighing balance, up to 4 decimal or 5 decimal, the you may say that of the weighing balances are available one, and it needs to be the calibrated one, internal as well as external calibration. You can have your the external calibration within a month, and internal calibration regularly. Instrument itself is doing its internal calibration one, whether it is okay, whether it is having some problems one, that all can be the recorded over there. Nowadays, this is not the sufficient thing. They wanted to make entry into that of the logbook also. How much of the compound they have weighed. You have to mention that, make an entry into their logbook, and what was the weight of that of the sample that you have taken. That also you should mention into the book. And another thing, that is, once you have mentioned it, you should have the printout for it, because when you are facing the audit, you are not just trying to see that is, yes, whatever the things I am sharing with you, that is all correct. They wanted to see the proof of it. And how you can provide the proof, that is, yes, this is the printout from that of the you must say that of the weighing machine. I weighed the sample at 10:30, and at 10 you must say that of the 31 I got the printout for it. But if I am getting the printout after the day after tomorrow or next day, they will not believe on it. Or they will say that you have to have that record one, the complete record, and at what time you will be there, and for getting that of the weighing and all that one, how you have prepared the sample, that all things they wanted to understand. The procedure that you are following for that of the analysis one, that also you should try to find out one. So first step I told to you or share with you, that is just weighing of the sample. You are weighing the sample, that is butter paper you are taking, how to cut the paper, butter paper. This is all skills. The people are tearing the butter paper with their hands. So this is not, you may say that of the proper way. You should use the scissor. You should try to make it into that of the square, and fold it so that you can, there is a the groove in between it, so that you can transfer the sample very easily one, or material very easily one. But if you are tearing the paper with that of your hands, there will be the threads along that of the edges one, and probably that may also interfere in your the result. So dear friends, at each step, your job and responsibility is very important one. You should understand your responsibility. You should try to know that to how much important you are in the pharmaceutical industry, and probably with that bend of mind you can have the lot of opportunity. With that bend of mind, you should go and do your the research work in the area of that of the quality control and that of the quality assurance. So step by step I'll try to go. That is I think method development I will try to share with you. That is how to develop the method. That is as you have weighed the sample. Second thing is that I wanted to know, that is a lambda max for that of the, if I'm working on that of the spectrophotometer, I can say that I have prepared the sample solution. It is a 100 ml solution. I have prepared it. Now I, with that 100 ml stock solution one, I wanted to go for that of the analysis related to it. Now with that 100 ml solution, you try to find out whether how you are trying to make a dilutions related to it. Now from that 100 ml, how much weight of the sample I should take, whether the drug that I am trying to have, or if I am having the API, I should take a weight, minimum weight, how much I should, whether I should take the 10 milligram or 20 milligram or 30 milligram. The question starts over here. And many times the students are getting confused with that, whether to take the 10 milligram of the sample or 20 milligram of the sample, drug one. And at the same time, they are confused that what volumetric flask I should use it. The size, what should be the size of volumetric flask, whether it is a, you may say that of the 10 ml volumetric flask, or 50 ml volumetric flask, or 100 ml volumetric flask. Dear students, try to avoid that confusion one. So minimum drug that you are, if you are having the scarcity of, or you may say that of the very limited quantity of the drug, then you can have the options one. But if you have the sufficient quantity of the drug, take 100 milligram of the drug in 100 ml. 1 milligram per ml, that is most important, you may say that of the dilutions you can have, that is a 1 milligram per ml stock solution, and from that 1 milligram per ml stock solutions one, you can make a necessary dilutions one, and with that dilutions you can come out with that of the analysis related to it. Where you can use that stock solution, now that stock solution can be used for that of the linearity studies one. Why there is a need for the linearity study, or that stock solution you can use for that of the stability studies also, whether that API is stable for the longer period of time or not, that we wanted to see. Now I wanted to ask you the very simple question. So am I audible, or it is just a one-way process? Hello, anybody can respond?
Yes, sir. Yes, sir. You are audible, sir.
Yeah.
Please.
So the very interesting thing that I wanted to ask you, that is if I am preparing the if I wanted to check the stability of the solution one, so how I am going to check it, how I can understand that solution is stable or not, if that is been exposed to that of the sunlight, and if it is having some sort of the degradations one, so it will not be the stable. But how I can recognize it? What are the options before me, how I can check it whether the solution is been stable or not? The first, the visual observations are important one. So you are trying to see whether the color has been changed, try to see for that first time whether the color is changed, whether you may say that if the color is not changed, then what changes are occurring over there, that you wanted to observe it. Yeah, and then second thing is that what you can do, that is take example of that of the you may say that of the lambda max of that of the drug. If you are taking the lambda max, if it is a shifting the lambda max, then you may say that of the, there is a possibility that the same drug is not there. The lambda max is shifting means what, some other type of the reaction or some reactions are taking place into that of the drug solution one, and it is not that much stable into the particular solvent one. Now, which solvent should be the use in that of the preparation of the stock solution one? Whether water, methanol, acetonitrile, or you may say that of the volatile solvent. You should not use the volatile solvent because it will change the volume. Again, there are many limitations. That is the you may say that of the classifications of the solvents are there. But at the same time, you can say that of the non-volatile solvents can be the preferred one. The water is a universal solvent, but as such, tap water you can't use. You have to go with that of the distilled water. Reason is that there is the presence of chlorides also. So that should be the avoided one. And instead of that of the tap water, you should use that is a distilled water one. In the case of your the HPLC, you can go with that of the double distilled water also. You can go with that of the double distilled water for that of the HPLC and for analysis one. Now, you may say that of the lambda max of the drug you can determine, and by which you can say that yes, there is a change in that of the lambda max, by which you can say claim that yes, the drug is not stable in the particular solution one or in the solvent one. Now, by this way, you can take a decision. So how to make the solution stable? What are the options before me? If the drug is exposed to the sunlight and if it is showing some changes one, then I have to protect it from that of the direct sunlight. I have to cover that drug or keep it in the dark for that of the longer period. And then I have to see it whether it is changing the lambda max. If it is not, then you can say that of the drug is not. You may say that of the stable drug is stable into the solution or in that of the solvent one for 5 hours or 6 hours. So this is what you can come out with that. That is a step related to that of the stability of the solution. The linearity also you can study. So this is also the one more step. That is the first step I can say that of the preparation of the stock standard solution. Second stage you can say that of the selection of that of the appropriate solvent for that of the solubilizing the drug. The third stage you can say that is yes, that is the once the drug is been soluble into that of the solvent one, whether it is a stable or not, that you wanted to check it. Then go with that of the lambda max determination for that of the drug. How much solution dilutions you can prepare? If you are having the 1 milligram per ml, then you try to make a dilution in such way that final solution will be you may say that of the 10 microgram per ml. So if or 20 microgram per ml you can take, and with that you try to check or try to see the lambda max for that of the drug. Once you have come out with that of the lambda max one, with the on that particular lambda max, if you are using the spectrophotometric method, you can go for that of the complete analysis related to drug. This is the stage that you are following. Next, you can say that of the linearity. And dear students, you have come across with that of the linearity study, with that of the Beer-Lambert's law. You have studied at your the third year or second year level, or in the final year level one. And with that, what is there in the Beer-Lambert's law? That is the two things are there. On one axis you are having the concentration, and another axis you are having the responses may be on that of the y-axis, and on x-axis you are having the concentration. So what should be the unit related to it? That should be the properly reflected one, whether it is microgram per ml, whether it is nanogram per ml, whether it is a milligram per ml, that should be written on that of the x-axis one. And on y-axis you can have that is a response means what? Absorbance. Response means what? Peak area under the curve. These are all the responses that you can have on that of the y-axis. Means what? You can plot the linearity curve in the range of, you may say that of the 5 microgram per ml to 35 microgram per ml. You are selecting certain things. Beyond that, it is not following the Beer-Lambert's law. That law you know, and you can understand that one. That is, as you are increasing the concentration, the response will not be the increase proportionately one. And you can understand that complete law accordingly. So you can say whether the drug is following properly linearity or not. How you can check it? By using the coefficient correlation value, that is the R-square value. And what equation you can draw from it? Suppose this is a mathematical thing now. You are having the straight line, and you know that when you are having the line in your the mind, you have come across with that the very simple equation when you learned the mathematics in the 10th standard or 12th standard, that is y is equal to mx plus c. That is called as the line equation. Slope and intercept. So y is the response. You may say that you have the slope and you have the intercept. The two things you are having, and the third things you can have the response. Then you can say that with the help of the slope and the intercept, you can calculate the concentration related to it. So it is possible for you. So linearity study is also the very essential component in that of your the development of analytical. This is all the steps I wanted to share with you, and these are all where your the skills are required. How to prepare the solution, how you are preparing it, and systematically you are doing it. And you may say that of the finally you are going for that of the analysis of your the formulation one, whether it is the tablet, capsules, oral liquids, whatever the things that you can say that you can make analysis related to. But try to understand how to, the important aspect is that how you are going to, you may say that of the how you are going to analyze, extract the drug from that of the particular formulation one. That is the challenging thing. And here your the skills are important, here your the knowledge is important, and here your the thought process is required. Many times the students are asking the question that, sir, how to follow the method if the formulation is a tablet formulation, if the formulation is a capsule formulation, if it is a, you may say that of the ointments and the creams, how to extract the drug from that of the particular formulation. The formulation is not been designed as per the wish of that of the analytical chemist. The formulation is been designed as per the requirement for of that of the patient, and it is not been designed as per the wish of that of the manufacturing person also, that, sir, this is a simple thing for me, so I will design only tablet formulation, I will design only capsule formulation. It is not like that. As per the wish, we are not designing it. As per the requirement of that of the patient, we are designing the formulation. If the patient is comfortable with that of the tablets, we are designing the tablets. And then it becomes the responsibility of we people whether the drug is stable in that of the tablet formulation, whether it is stable in the oral liquid formulation. If it is not there, how I can make that drug stable into that of the formulation, that becomes my responsibility. That becomes the responsibility of the analyst, or you may say that of the chemist who is working in that of the area one, how to make that one. And in some part of it is there in that of the R&D sections. Some part of it is there in the R&D section, that is research and development section one. And here, that is how to make it stable one. There are the different scientific approaches one by which you can make that particular formulation stable. It is up to you. You can have the thought process related to it, you can have the literature related to it, and you can have the many things which may be useful to you for that of the study. So dear students, then when you are making that formulation the stable one, as I am talking about, that is the extractions of that of the drug from the formulation. Take example the tablets. We are taking the 20 tablets for that of the dissolving it into that of the certain solvent, sonicating it for that of the 10-15 minutes, and making up the volume up to that of the 15. Now I wanted to ask you, that is how much tablet you should take. Why only the 20 tablets? Why not one tablet? Why not 2 tablets, 3 or 4? Why only you need the 20 tablets? What is the reason behind it? Have you thought about it? And why in the analysis one, the readings should be the repeated for that of the 5 or 6 times? Why they are not satisfied with that of the 1 or 2 readings only? That is, yes, the 2 readings are sufficient one. It is not there. You have to repeat it minimum for that of the 5 readings. Why it is so? Because they wanted to check how much errors are there. And for checking the errors one, the standard deviations, or you may say that of the percentage RSD, the statistical parameters they are applying, and with that statistical parameters they wanted to check whether that formulation, whether that readings are stable or not, whether that it is following the as per the expectations of that of the you may say that of the standards one or not, that you can check it. So these things are generally been are there in that of the related to that of the formulations one. When you are checking it, you have to apply that of the statistics related to it. And in the extraction case, as I told to you, that is the 20 tablets, or you may say that of the white is required. If it is a you may say that of the ointments and creams, how you are extracting it? How, what are the methods you are trying to use for the extraction of the drug from that of the ointments and cream? In the form that of the pharmaceutical, that is the capsules one, or you may say that of the oral liquids one. And in the particularly in the case of oral liquids, you should know the density of that of the oral liquids one. And likewise, you can you may say that of the do your calculations one and the readings one. Otherwise, every time you are taking the weight of the sample, that is not sufficient. How much quantity should be taken? You refer that pharmacopoeia for that. So dear students, whatever the things that you are doing in your the laboratory, that is a practice, that is a rehearsal for you to go to the industries one. There are huge and ample opportunities for you in the industries. Once you have come out with that of the development of the analytical method, you have, this is the role for you. Now glamorous things in the analysis is that, have you worked on the HPLC? Few students may have worked on the HPLC, high performance, or initially it was called as a high pressure liquid chromatography. Now it is called as a high performance liquid chromatography. So when you talk about the HPLC one, you should know, if I wanted to work on that of the HPLC one, what skills am I going to have? What skills are required? So if I'm talking about the HPLC one, I should know that is how to operate the system one. That skills are required in the industry one. You learn HPLC in your institute one. When you will go to that of the MFARM QA or MFARM analysis one, the instrument will be given to you, dedicated instrument will be there where you can work on that of the instrument and you can acquire the skills related to it. How to operate it, whether what is a gradient system, what is the isocratic systems one, what different types of the pumps they are using it. Then you may say that of the purging, what does it mean? Purging means what? What are the tubings they are using? What type of the columns they are using, whether it is a C8 column, C18 column, or you may say that of the what type of the detectors they are using it, whether it is a you may say that of the UV visual detector, photodiode array detector, RI detector one, or you may say that of the mass as a detector, which type of the detectors they are using it. So from my point of view, HPLC, even though you find that it is a very glamorous one, HPLC, HPTLC, GC, these are all glamorous instruments, but you talk about the HPLC, it is a column chromatograph. What are the different things available into it? That is the first important thing is that you are having the reservoir. Second, that is the tubings are necessary. The third, that is you are having the pumping system. The fourth, you are having that is the columns one. And the fifth one, you are having that is the detector one. Prior to columns, you are having the injectors. That is the injector port. And with that injector port, the limited volumes of that of the sample you can inject, you can try to find out the responses one. Responses means what? It is a chromatogram. Responses in the HPLC, it is called as the chromatogram. So in the quality control, in the quality assurance one, what is the need? That is, there is a need for that of the trained manpower who can operate this HPLC system, who can operate HPTLC, GC1. Now in your MFARM, there will be a lot of opportunities for you, that you are having an opportunity to learn this instrument, to get acquainted with this instrument, and then you may say that of the industries, in the most of the industries one, even if you are naming that is a big industries one, you will find minimum 150 or 200 HPLC systems are there in the industries one. And who are operating this one? That all are operated by that of the quality assurance people or quality control people one. So the opportunity for you there in the area of quality control and that of the quality assurance one. Further, you have the opportunity for that of the in the method development, validations of the method, that is also there. Regulatory, in the case of the regulatory also, you can have the opportunity. Further, after you are the B farm, you can go for that of the M farm in the quality assurance. There are now the specific branch analysis, pharmaceutical analysis is also there. Then you can say that of the you can have the opportunity after your M farm can go for the PhDs. Even in the abroad you can go, and the research area is the vast area where you can have the ample opportunity to do the research work. In the academic institute right now in India, you will find many central institutes one. Many university departments are there. Private colleges are also there. Private universities are there, where there is a need of that of the trained people who can operate such type of the system, who are having the skills to operate the instrument one, who are having the knowledge for that of the research one, and they can contribute significantly in the area of the research one through their knowledge, publications of that of the research. In the even the patents also, you have a lot of scopes. You can contribute in the patents also, because when you are submitting the data of patent, they will ask you that is whether you have analyzed this one and what methods you have adopted for that of the analysis of that of the particular the sample one or the drug one. So you should know that is the analytical method. So your job and will be there, more jobs will be there for you. And these are all, you may say that are the cream positions in the industries one. Another important aspect I wanted to say that if you are not that much happy with that all these things, then you can go for that the one more area of the research one, I can say that is the forensic science. And in the forensic science, Sherlock Holmes, the story of the Sherlock Holmes, you may have studied one, the investigations and how they are investigating it. That is, suppose there is some drug addiction, there is some sort of the drug abuse one, there is some sort of the poisoning one. They wanted to check that is whether the patient who has consumed that particular chemical, they wanted to identify it. How they are identifying it? By using all these sophisticated instrument one. So dear students, you can learn that all these instruments, whether it is a HPLC, LCMS, whether it is a GC, HPTLC, these are all instrument technical skills you can have at your the postgraduate level or at your the PhD level. So scope for you is a tremendous scope. You have the opportunity to go to abroad. Even in the abroad, the people are looking for that of the very experienced people, expertise people who can work on that. You can have the opportunity in the research area. Right now, the Indian government has given the scholarship for the PhD around 40,000 per month, 35 to 40,000 per month for that of the project research fellow. Now try to see, the industry is giving that 25,000 rupees per month or 30,000 rupees per month, and the scholarship for the PhDs they are having, that is the 35 to 40,000 rupees per month one. Plus they are having the opportunity to go to abroad for the presenting their research work, for sharing their research work. At the same time, they can, after their M.Pharm one, they can go for that of the PhDs, they can do their research work and can go to the abroad for that of the further research also. Or they can stay in India, and there are a lot of opportunities for them. So in the quality control section particularly, or in the analytical section, I can say at initial stage you can be the quality control officer, then senior officers related to it, quality control or quality assurance manager one, senior manager will be there, head of the departments, director, and vice president of the company. So you can be in this segment, the top position is that of the vice president. So I wanted to say that when the students are trying to achieve these positions, one, at least to reach to that of the vice president level, the duration will be required, that is the 15 years. Within the 15 to 20 years, you can reach to this position with a package of 60 lakhs, 70 lakhs right now. But when you will join that one, and after your 25 to 15 or 20 years of experience one, that package will be 1.5 crores rupees. Because every time when I am discussing with the students, my students one, they are talking about that of the packaging one. What type of the package I will get in the industry one. So initially that package may be the less, but as you are entering into the industry one, you are at each level, you are getting an incremental value. And finally, when you go to that of the vice president level one, it will be around, you may say that of the 60 to 70 lakhs. Big industries are giving this much one. Now, this is what the glamour one. This is what the challenges one. This is what the, you may say that of the all the things are required. Here the skills are required, communication skills are required, technical skills are required, the presence of mind is required, and how you are taking your industry forward, that is also required. Even you can start your own industry where you can analyze the API samples, where you can synthesize the impurities one, you can analyze that impurity, and you can supply that impurities to that of the pharmaceutical industry. This all could possible, can possible to you if you are doing, you are the masters in that of the, in the area of, I can say that of the pharmaceutical analysis, pharmaceutical chemistry, or you may say that of the quality assurance, or quality control, or quality assurance. So these are the promising areas for you. I wanted to say that, I don't wanted to say that some other areas are not important. I will speak equally on that of the same area, because it is a integral approach one. I can't say this is the big one, this is, but you have tremendous opportunity to work in this area one, and there is a huge requirement for that of the in the pharmaceutical industry. This is a, you may say that of the as we call the India as that of the medicine of the world. So because of that, you have the lot of opportunity, tremendous opportunity, and the people, those who are there in the area of Mumbai, Pune, you may say that of the Baroda, Ankleshwar, Ahmedabad, in that region, you will find that this belt, Thane, and you may say that of the Navi Mumbai also, will find the huge number of pharmaceutical industries, and their requirement is more. Even the students from that of the different schools and colleges, they are moving to that of the industries one. Still, there is a big requirement. So, dear students, only thing is that the two things you have to study very seriously. That is, you have to acquire the soft skills, and the second, that is the technical skills. And at the same time, you need to have some sort of the higher qualification. For having this type of the, you may say that of the glamorous the positions one, you can't go with that of your the B.Pharm. You have to go with that of the M.Pharm, with that of the PhDs one. That is also necessary. Plus, I can say the MBA, if you can have, and the addition to it, probably it may in the long duration may be helpful to you. But the technical skills, I can say, if you are doing your the M.Pharm one, that is it will be helpful to you for your the getting the jobs in the industries and getting some the crucial positions in that of the industries. So one more slide, or one or two slides I will go, and I will conclude the speech over here. So these are all the areas, you can say, the bioanalytical method, health in needs, medical transcription, regulatory affairs, forensic science, scientific writing, and academics. In all that areas, when you have an opportunity. But what qualifications is required? Minimum qualification will be, that is a B.Pharm is not sufficient, but you need to have the masters or that of the PhD related. And in the particular area of, you may say that of the pharmaceutical analysis or quality assurance. Okay, so I hope these are all the areas open for you, and they are waiting for you. That is, only thing is that you have to acquire certain skills and the knowledge one. So thank you very much for patience listening, and if you have any questions, if it is possible for me to answer your queries one, I'll be happy to answer your queries. I wish you all the best, and I'm really thankful to Vivekananda Education Societies College of Pharmacy for organizing such wonderful, the you may say that of the online, the session one on the very important topic of that of the quality assurance, or you may say that of the analytical sections, which is very important in the pharmaceutical, the industry. The thank you, Ojas. Thank you, Mustaq sir, uh, for this all arrangement that you have made over here. Thank you. So over to
Thank you
you.
very much, sir.
Yeah.
Yeah, sir, definitely. I was very much present throughout your
Oh,
talk, and I could say, sir, you are a avid teacher. A teacher is one who does not just only teach, he also shows us a path. And you started your talk with showing us a path in the words of Swami Vivekananda and Martin Luther King Jr. Sir also deliberated on with the various department in industry and how the QA QC connect to them. What is the significance and what actually this department will do. Sir also explained that to student, and I am very sure student have taken a note of all the points sir has mentioned. Sir, we were trying to look into if there are any questions throughout your presentation, but it seems
okay.
everything is crystal clear to students. They have understood it very well. And anyways, this presentation is going to be available. They can refer it anytime. Sir, we also talked about quality of raw material, then how the formulation will go. Uh, that no product or formulation will go without QC department looking into it. We also said not only about
Yeah.
QA or QC area, we also talked about research in what is it? Actually, it is name of giving some solution to the problem, not creating new problem, and also deliberated on research steps involved in kind of research process. And in the meantime, you also give your personal input, which you must have learned about 25 years of your experience. We also talked about importance of simple things like what we learned in undergraduate level, calibration and different parameters, pressure, temperature, everything. Uh, that the one part I really liked was, sir, that you talked about the skills required, both soft and hard skills required for a person if they are really interested in the field of QA and QC.
Yes.
Uh, and the last part, student might have really liked that what are the career avenues. You talked about that what department like regulatory and packaging. These are two department which they can think of other than being in just research. Then you also showed them the way of abroad studies and what are the scholarship government and available throughout the world. And of course, PhD and MBA, these were the two areas the student must have noted down, and they would have understood that learning is not going to aid at least at UG level or PG level. Sometimes you have to even think beyond it. So you have to always be a student. Thank you very much, sir. We were thinking that if somebody would have any question, but unfortunately or fortunately, there are no questions. Students have taken all the things in positive sense. So I would request now Ojas Agarwal sir to kindly place an vote of thanks. Ojas sir, over to you.
Yeah, thank you, Mustaq sir. Sir, I would like to express my heartfelt thanks to Dr. Atul Shirkhedkar sir. Sir is currently the Vice Principal, Professor and Head of Pharmaceutical Department at R.C. Patel Institute of Pharmaceutical Education and Research, Shirpur. Sir, thank you very much for sparing the time from your valuable time from your schedule and enlightening the students regarding their path, career path in MPharm and the research related to the Master of Pharmacy. I would also like to express my heartfelt thanks to VES management and Dr. Supriya Sidhe, Principal, VES College of Pharmacy, for allowing us to conduct such type of the online series. At the outset, I would also like to thank Dr. Rajashree Hirlekar Madam, Head of the Department Pharmaceutics, Dr. Mustaq Sheikh, Head of the Department Pharmaceutical Chemistry, and Dr. Anita Aire Madam, Head of the Department Quality Assurance, for giving me an opportunity to propose a vote of thanks to my beloved MPharm Research Guide, Dr. Shirkhedkar sir. Thank you very much.
Thank you.
Dear
Thank
sir, it
you.
was an absolute pleasure for us to have you today morning over here, and I must ensure that the student enlightened with the thought and the views that you have expressed very the politely, and the future prospect that you have given them, starting from their basic research career to the managerial level or the deputy managerial level. We would like to have such kind of the kind cooperation in future as well, sir. Sir, this is an online platform, but definitely we will plan some offline activity also, so that we'll call you for that particular kind of the thing as well, sir. Thanks to all
Thank,
participants and the viewers over here. I am sure that they must have gained the guidance related to the higher education and the future perspective with the pharma industries. With this, I would like to conclude this particular Research Connect series session. Thank you very much, sir, and thanks to all participants. Thank you
thank
very
you.
much.
Wish you all the best. Yeah.
Thank you
So
everybody,
let's stop
sir,
here.
Ojas sir,
Thank
and
you
the
very much.
participants, and the ICT team, which was at the
Yes.
back end helping us
The
throughout.
best, the support extended by the ICT team also is awesome. Thank you very much.
Yes. Few announcement at the end that Research Connect series was pointed out to help the research enthusiasts to take right career choices through this series of seminars and workshops. So we have conducted 2 seminars till now and 1 workshop. We had asked students to invited students to come down to college, and we had showed them various instruments and how research flows from one department to other department and how it is multidisciplinary. So, with looking to the fact that this thing is really important before you choose out your areas of research, uh, we have now announced that on 7th and 8th October, we are rolling down phase 2 of our Research Connect workshop, where all the BPharm graduate students, they are invited to attend this session and get the real hands-on, and also understand that aptitude of yours. When you will be in the lab, you will really understand whether you like the field or not. So it will help you to choose the right, I'll say, kind of a stream for you and career for you. With this, I thank you everybody, and
Thank
I announce that this seminar is concluded. Thank you very much.
Thank
Keep
you.
Thank
connected.
you, sir. Thank you, sir. We
Yeah.
are logging off
Take
right
care
now.
of. Yeah.
Thank
Thank
you
you.
very much.
Shall I leave?